Hi everyone, we are continuing with the topic 2 of human anatomy class, and this is part 2 of the topic 2 lecture. We've already covered the plasma membrane and now we're going to start with cytoplasm. Cytoplasm is the material that is present inside the plasma membrane and it is outside of the nucleus. The nucleus is one of the most important organelles inside the cell. And the cytoplasm is made up of three components. The first is the liquid part of the cytoplasm, which is the cytosol, and it is also known as ICF or intracellular fluid. The second component of the cytoplasm is the little organs or the organelles, which is actually the metabolic machinery of the cell. It carries out all the functions of the cells. And the third component is inclusions. Inclusions means anything that the scientists could not include in the cytosol or organelles, they've pooled all of that into the organelles. So they include chemical substances, stored nutrients, or cell products. This table lists the structure of a representative cell. So on top is the plasma membrane and the cytosol, followed by a list of those organelles which do not have a membrane covering around them. That means the non-membranous organelles. And the table continues with the list of membranous organelles. That means organelles that have a membrane covering around them. This figure shows the anatomical structures present inside a representative cell. This slide gives a list of the membranous organelles inside the cell. All of these organelles listed here are covered by a membrane which has two layers. So it's called a double membraned organelle. And the functions of these organelles are also provided. We will start with the nucleus. The nucleus is a membranous organelle. It is considered to be the metabolic center or the control center of the cell. The nucleus is surrounded by a double-layered nuclear envelope. All the material that is inside the nuclear envelope is considered the nucleoplasm. The nuclear envelope is interrupted by pores or holes which are known as the nuclear pores. So things can move out of the nucleus and into the nucleus through these pores in the nuclear envelope. Now, because the nuclear envelope has two layers, there is a space between these two layers, and that space is known as the perinuclear space. Now, if you look inside the nucleoplasm, you will see some long thread-like structures which are known as the chromatin or the chromosomes. These contain the genes. So these are the reservoir of your genetic information. In addition to chromatin, you will also see some very dense circular structures which appear to be very darkly stained under the microscope. Those are known as the nucleoli. If it is singular, it is nucleolus. If it is plural, it is nucleoli. So these darkly stained or densely packed structures or the nucleoli are very important because these are the site of ribosome synthesis. What are ribosomes? We will learn about it in the later slides. This multi-panel figure shows all the components of the nucleus that we have just described in the previous slide. Here is more information on the nuclear membrane or the nuclear envelope, the nucleoli and the chromatin. Now, something interesting about the chromatin is that the chromatin on which your genes are present, it is made up of DNA. DNA is your main genetic material. It stands for deoxyribonucleic acid. And in addition to DNA, it also contains a specific protein, and the name of that protein is histone. And the way the DNA and the histone proteins are arranged, it's known as beads on a string structure. And this structure is called nucleosome. This figure shows the beads on the string or the nucleosome model. Now, what happens when the cell is not dividing? The genetic material stays in the form of chromatin, which is long, thin, thread-like structure. When the cell is undergoing cell division, right before it enters the cell division stage, the chromatin material, it condenses to form shorter, thicker structures. At that time, we call it the chromosome. If you look at this figure, it's also a multi-panel figure. The top right part, it shows the nucleosome model. There are 8 histone proteins which look like beads, and the DNA, which is a string-like structure, is coiled around these histone proteins. The next organelle is the mitochondria. The mitochondria are considered to be the powerhouses of the cells. Why? Because this is where energy is generated for the cell. Cell needs energy for all of its functions, and somebody has to produce that energy, and it is the mitochondria. How is the energy generated? Energy is generated from food, and oxygen helps in this process. So oxygen is used to break down food, and a huge amount of energy is released, and that energy gets stored in the cell in the form of ATP. ATP stands for adenosine triphosphate. So these ATPs are energy rich molecules, and whenever the cell needs energy for a specific function, the ATP is broken down to release the stored energy. This figure shows the anatomy of the mitochondria. The mitochondria are membranous organelles. They also have double-layered membrane. The outer layer is smooth. The inner layer has infoldings, and those infoldings are known as the cristae. On the cristae, there are special proteins which are enzymes. Enzymes are biocatalysts. That means they help to speed up chemical reactions. And these enzymes are used to break down food, organic molecules, and using oxygen. And the end product of this reaction is carbon dioxide and ATP, again adenosine triphosphate, which are the stored energy source. Continuing with the membranous organelles, the third one is the endoplasmic reticulum, or the ER. The ER is a network of thin tubes which is hollow, and it contains a fluid inside it. It is continuous with the nuclear envelope, and there are basically 2 types of ER. The first type of ER have ribosomes attached on their surface. So if you look at them, they appear to be rough. So those are known as rough endoplasmic reticulum, or the RER. The other one does not have ribosomes attached onto its surface, so it appears to be smooth, and that's why it is known as smooth endoplasmic reticulum, or SER. The rough ER, the main function is protein synthesis. Why? Because ribosomes synthesize proteins. Now, if you remember from one of the previous slides, where were ribosomes synthesized? Inside the nucleolus of the nucleus. The smooth endoplasmic reticulum, on the other hand, are involved in metabolism of fats and also in detoxification of harmful substances. The top panel of this figure shows how the endoplasmic reticulum are in very close proximity of the nucleus because they are continuous with the nuclear envelope. Now, if you look at the left bottom panel of the figure, it shows you both types of ERs, the rough ER with the ribosome on its surface and the smooth ER with no ribosomes on the surface. Functionally, the ER is the main folding and packaging center. So, whenever a protein is synthesized by the ribosome, the protein moves inside the lumen of the endoplasmic reticulum. There, the protein gets folded, and refolded, and then it is stored in a small vesicle which is known as the transport vesicle, and the transport vesicle gets pinched off from the endoplasmic reticulum, and it travels through the cytoplasm to its next destination, which is the Golgi apparatus, because the Golgi apparatus is the processing center. Now imagine you have bought a fragile gift for a friend who lives out of town or out of state. That means the gift has to be sent to the friend via mail. What would you do? You would try to package it properly so that it doesn't break during its journey. So you might buy a bubble wrap or something, a good package material. So that is exactly what the endoplasmic reticulum is doing. It is packaging the protein, making it secure. So the Golgi apparatus is the next membranous organelle. This is the main packaging center for the cell, processing and packaging center, and it produces different kinds of packages. It could have a secretory vesicle, a vesicle for the cell membrane, or even a lysosome, which is another organelle inside the cell. Now remember the fragile gift that we were discussing in the previous slide. So now that you have bubble wrapped it, what would you do? You would take the gift to the post office because it has to be mailed to your friend. So the Golgi apparatus is that post office. The protein, after coming out from the endoplasmic reticulum, it reaches the Golgi apparatus where it is processed further, and then depending on where that protein is needed, Golgi apparatus will package it and send it accordingly. So if you look at this figure in pathway 1, the Golgi apparatus is packaging one specific protein, and these proteins are to be secreted out of the cell. So a vesicle forms in the Golgi apparatus, which is called a secretory vesicle. The vesicle comes and fuses with the cell membrane, and then the contents of that vesicle, they get exocytosed out. That means thrown out or kicked out of the cell. Now if you look at pathway 2, there is a particular protein that is coming, which would be forming a component of the plasma membrane. So that secretory vesicle comes, and the secretory vesicle fuses with the plasma membrane, and the components are used in the repair or maintenance of the plasma membrane. Now going to pathway 3, here the proteins that came out of the Golgi apparatus were very strong digestive enzymes, and these enzymes would become part of the lysosome, which is another organelle. So the Golgi vesicle containing these enzymes would go and it would fuse with the lysosome so that the lysosome may get all the materials that it needs. So what are lysosomes? Lysosomes are sac-like structures present inside the cell. There could be more than one lysosome in a cell, and the lysosomes contain really strong enzymes which are hydrolytic or digestive in nature. That means these enzymes can be used to digest or break down anything that is not required or the non-usable materials in the cell. Sometimes even organelles, if they have become damaged or they have become really old, those can also be broken down and recycled by the lysosomes. Any foreign material that gets inside the cell, the lysosome enzymes, they lyse that foreign material or break it down. Every now and then, if the cell has become senescent, that means the cell has become old or the cell has been damaged beyond repair, so then what happens is all the lysosomes inside the cell, they rupture and they release all of the strong digestive enzymes, killing the entire cell. And this phenomenon is known as autolysis. That means self-breakdown. Peroxisomes are organelles that are very similar to lysosomes. They also contain strong enzymes which can be used to break down or lyse unwanted materials. They mainly detoxify or break down hydrogen peroxide, alcohol, and formaldehyde. See the similarity between the peroxide and the peroxisomes, so please remember this. They convert this harmful hydrogen peroxide to something harmless such as water and other materials. We are done with the membranous organelles, so we move on to the non-membranous organelles. These organelles do not have a membrane covering them. The first one is ribosomes. The ribosomes are synthesized inside the nucleus in the nucleolus, and there are two types of ribosomes: free ribosomes and fixed or attached ribosomes. The free ribosomes are floating freely in the cytoplasm, while the fixed or the attached ribosomes, they attach to the surface of the rough endoplasmic reticulum. Both the types of ribosomes are involved in protein synthesis, that means making proteins in the cell. The second type of non-membranous organelles are the microvilli. The microvilli are really small, finger-like extensions or projections of the cell membrane. Their function is to increase the surface area of the cell membrane. Why would cell membrane need increased surface area? To enhance secretion and absorption. Moreover, as the microvilli bend, back and forth, it's a wave-like motion, It helps to circulate the fluid outside of the cell, which is known as the extracellular fluid, and this movement helps the cell to absorb more nutrients. Cytoskeleton is the next non-membranous organelle. This is the skeleton of the cell. So if it is a skeleton of a tiny cell, it is actually a really thin thread-like structure, and these thread-like structures are protein fibers which are spread all over the cytoplasm. The cytoskeleton helps the cell to maintain its shape. It also helps the cell to move. There are three types of cytoskeletal elements: microfilaments, intermediate filaments, and the microtubules. Microfilaments are the thinnest cytoskeleton. Their diameter is about 7 nanometers. The intermediate filaments are slightly thicker than the microfilaments, and their diameter is around 10 nanometers. The microtubules are the thickest cytoskeletal elements present inside the cell, and their diameter is about 25 nanometers. This figure shows pink-colored microvilli, or the finger-like projections, and the shape of the microvilli are maintained by microfilaments, which are the cytoskeleton which are present inside the microvilli. Now, if you look in the same figure a little bit down, then you can see the intermediate filaments and the microtubules. The microtubules, which are the thickest of all the cytoskeleton, they form three important structures inside human cells. Those structures are centrioles, cilia, and flagella. Centrioles are important for cell division, and we will learn about it later on. The cilium, which is singular, the plural would be cilia. These are short hair-like structures present in some of your tubes inside the body, for example, the trachea. The flagellum is the tail of the sperm. The microtubules are arranged in a specific fashion inside the centrioles and the cilium and the flagellum. In the centriole, the arrangement is known as a 9+0 array. If you look at panel A of the figure here, it shows you the arrangement of microtubules in the centriole. The 9+0 array means in the periphery, the microtubules are arranged in triplets, and there are 9 sets of triplets. And in the center, there is no microtubule, so 0, 9+0, 9 triplets arranged in the periphery and 0 or nothing in the center. Now, if you look at a cilium or a flagellum, the arrangement of microtubules here is a 9+2 array. That means there are microtubules, again 9 arranged in the periphery, but these 9 microtubules, these are present in doublets, not triplets. And in the center, there are 2 microtubules. So it's a 9+2 array.